So in your book, you talk about golden cells. What are golden cells, and what is your process for selecting cells for treatment?
Okay, well, so the golden cell thing came about in 2010. We actually had a patient from Dallas he's a 3-time state champion high school football coach in Texas. That means he's sort of way up there, and particularly for the people in the community. And he had crippling MS. He was in a wheelchair. He had been essentially decommissioned, and he was on disability. And the community basically rallied around him and said, "You need to go to Panama and try these cells." He came down, and his name is Coach Sam Harrell, and there's probably a lot of stuff on the internet. His son was actually an NFL player. His son, I think, is a coach now. But like NFL, is he an NFL coach? Yeah. So anyway, Sam comes down, and he gets this incredible response, right out of the chute, out of his wheelchair in a month, and exercising and all this kind of stuff. And that lasted for about 14 months, and then he started coming back down again. So then he came back for treatment, and he got a little bump, and historically, his history has been hijacked a couple times on this, but we do have data that he did improve somewhat after the second treatment, but it wasn't near the benefit that he got from the first treatment. Then the third treatment in 2012, boom, it was like the hand of God touched him, and there are videos of him run up downstairs, and basically he went from being completely disabled to now he's back coaching at the same place, and here we are, 14 years later, he's back coaching at Ennis High School, where he was, state champ for all the time. So I'm curious, and I was like, well, what's the difference between 10, 11, and 12? Pretty obvious question, isn't it? So, and Dr. Paz mentioned that we have samples of we always hold back samples, and we're running into a storage problem now because we have so many samples from so many treatments, but we keep some from everyone. So I called them golden cells for lack of a better term. I said, look guys, we have these miracle cases, then we have other cases that aren't so miraculous. Let's find out what the difference is. So I went to all the doctors, and we sat down, and we said, all right, give us your top miracle cases. This was in 2012, so we got a list of 20 patients that were just like surprisingly, crazily benefited from the treatment. And so we selected, and of those, there was commonality in one of a couple of the cell lots. So we took those cell lots. We took 6 cell lots, meaning a cell lot can treat hundreds of people, and new technology has allowed us we can treat millions of people from a single lot. But back then, we could treat hundreds of people, and so we tested. I'm trying to not go geeky scientist on you here, but we basically took those cells and we grew them up, and we grew them in 2 dimensions, and we grew them in 3 dimensions, because we know that ultimately you're going to have to grow in 3 dimensions. So we grew them up, and the whole process took about a year until we had all of the cells grown in 2 dimensions, 3 dimensions. We took those golden cells, or those cells that had golden effects, and then we took cells that didn't have such great effects or mediocre effects, and then we took fibroblasts, which they're healing cells, but they don't secrete a lot of the stuff that the MSCs do. And then we ran an analysis of what molecules were in there, and it's amazing. If any, I know, who's a fan of statistics in here? Nobody. No, well, a couple of, I got you. No, I just kidding. So, a p-value, which is the probability of something being chance. Typically in science, we look at a minimum of p05, so that'd be the difference between this data set and this data set, and that's a 95% chance that it wasn't chance. And if it's a p01, then there's a 99% chance it wasn't chance. And if it's a p001, it's a 99.9% chance. So the more zeros before the 1, we had, of some of these molecules, they were 14 zeros before there was a single 1. And this was the difference between golden cells and I guess, middle of the road cells. So starting that, right then, we started screening every donor for the presence or the absence of these different markers, and then we were able to screen. And so from 2000, really, I think it was probably 2013 when we got it all finished, and we developed the tests. Then from then on, we've only used golden cells. So before it was crapshoot because we didn't know what we knew, and then once we learned that, we instituted we're only going to use golden cells. So we throw a lot of cords away, which is expensive because you wind up, you already have the expense of isolating them and everything. You have to do a certain amount of work before you can test them. And so that's what I call golden cells.